we have, as Werner already pointed out, we have a working group in Committee 1 on individual radiosensitivity, given the strong interest in this topic. And the chairperson of this working group is Preeta Rajaraman, who unfortunately was not able to come. So I will present this topic. We have been discussing, of course, the content of the lecture, but and it's to a large extent, let's say, my point of view. We do agree generally, but you will see it. My intention is to provoke discussion a little bit. It's a controversial topic, I would say. It's not straightforward to identify radiosensitivity. Well, I hope you will see it. There are some reports that you can look into. A very comprehensive report published by Public Health England, at that time Health Protection Agency, in 2013, and there is a report from ICRP, publication 79, on genetic susceptibility to cancer. Now, one thing up front is that there is quite a lot of controversy or misunderstanding when it comes to the term radiosensitivity. So it's very important to define what do we mean. So you can talk about whole organism radiosensitivity. You can talk about normal tissue radiosensitivity with respect to side effects of radiotherapy, so clinical radiosensitivity, normal tissue radiosensitivity, non-cancer, non-deterministic effects, susceptibility to radiation carcinogenesis, tissue radiosensitivity for cancer, or cellular radiosensitivity. I'm not going to talk about everything. I will focus on these 2 points that are marked with the arrows, and there has been a very nice paper published by Nicolas Forêt last year in Mutation Research that some of you may know, where he introduced some terms in order to avoid misunderstanding. So we stick to this terminology. So when we speak of radiosensitivity, we think of the organ radiosensitivity towards radiotherapy. So when we expect, if a radiosensitive patient is at a higher risk of developing side effects, deterministic effects to radiotherapy. Well, when we speak of radiosusceptibility, then we understand this as the higher risk of developing radiation-induced cancer. Now, an example why it is very important to distinguish the different forms of radiosensitivity is that, of course, we all agree that children are radiosensitive. And you see this on the left panel. We have this these are results from the LSS study. But if you go out to medical doctors, to radiation oncologists, and you tell them a child is radiosensitive, they will say, what? A child is very radioresistant because I can treat a child with high doses. He will develop some side effects, but they will heal immediately. Very radioresistant. Yeah, so children are radioresistant with respect to deterministic effects, but they are, of course, very radiosensitive with respect to stochastic effects. And you see on the right panel, this is a study on mice, not on humans, of course, where you can see the medium lethal dose that is needed to kill 50% of animals, and you see that this dose decreases with age. So young animals are very radio resistant, again, with respect to deterministic effects. Now, I think most of us assume that high radiosensitivity and radiosusceptibility, now I cover both, this is an intrinsic trait. So I am intrinsically radiosensitive, or I am intrinsically radio-resistant. And the reason why we assume this, and we generally, you hear often that the people are differentially radiosensitive. The reason why we say this is that we know that there are approximately 15 diseases, known diseases, where people who have these diseases are radiosensitive, and you have a list of these diseases here. So they are radiosensitive. Some of them, at least, I'm not listing all 15, and some of them are also radio. They are also they have a high radiosusceptibility. What is interesting is that there is obviously not always a correlation between both. You see, so these diseases, for example, the patients are they are radiosensitive, but they are not radio susceptible. Oh. So this is worth keeping in mind, that there is not necessarily a correlation between radio sensitivity and radio susceptibility, which again emphasizes why you have to be very precise when you define. Well, I mean, you have to define what you speak about. These diseases are rather rare, so there are very few people who have them. They are the result of low frequency, high penetrance mutations. Okay, and now if we assume that we are intrinsically radio sensitive or intrinsically radio susceptible, then we can take tissue samples, let's say peripheral blood lymphocytes or fibroblasts from a person, and determine the cellular radiosensitivity of these tissue samples. So take lymphocytes, irradiate them under in vitro conditions, and look for DNA damage, chromosomal aberrations, survival. And if the world would be beautiful and simple, and we were all young and rich and beautiful, then this is what you would get. And this would be an ideal outcome. You have a very good correlation between radiosensitivity and radiosusceptibility and the cellular radiosensitivity. And there is a lot of publications, a lot of attempts to find this correlation. And there are different assays that people have used. And these assays, you can put them together and call them functional assays, because you irradiate cells and then you look at the net outcome in terms of survival or in terms of chromosomal aberrations, or how they repair DNA double-strand breaks, for example, which we know that are very important for what happens to a cell that was exposed to radiation. And then we can also look at gene expression profiles. We can look at differences in single nucleotide polymorphism, so-called SNPs. We can look at individual SNPs, or we can look at all polymorphisms in the genome, and this is called GWAS, genome-wide association studies. Now, this is why we do this. That's a nightmare of a radiation oncologist. So you treat a patient, like this patient was treated for Hodgkin disease, and 6 years later he came back with this symptom, and you think, wow, he must have been extremely radiosensitive. We got blood from this patient and we didn't find any increased or any phenotype of radiosensitivity. We looked at chromosomal aberrations. I think we looked at foci as well. I don't remember anymore. So why did he develop these side effects? In fact, he was a physical worker, so he did some physical work, and as far as I understand, that's what he said. He strained himself. So this is probably the outcome of physical exercise or physical work. Probably nothing to do with high radiosensitivity. But this is a patient where you would think, well, yeah, this is the guy. We're going to look at his cells, and he must be radiosensitive. Not necessarily so. And if you look at, as I said, there are very many results or publications where people were looking at functionality assays, because this is very appealing. You know, you collaborate with the radiotherapy department, and they treat many patients, and of course they will have patients that develop side effects. So you take blood from these patients, you take blood from patients that didn't develop any side effects, and then you run a functional assay. And the outcome is extremely controversial. So there are publications where a very good correlation is found between in vitro radiosensitivity and clinical radiosensitivity, like these two papers. Residual 53 BP1 foci counts, which were counted 24 hours after in vitro radiation, were significantly higher in fibroblasts from radiotherapy-sensitive patients. But then, and similar here, but these were different foci, and they were looking at peripheral blood lymphocytes. But then you have publications where there is no correlation whatsoever. So I think if you, and I'm not going to show you a table with all publications, there are really many. But it seems like there is no clear outcome. I think generally one can say these assays don't really give a clear image. They don't give a clear result saying, yes, there is an assay which is predictive. We don't have an assay like this. It's actually quite disappointing. Now, what about genome-wide association studies? We are all different, and we vary. Although we are in the same. We belong to the same species. So why do we differ? We are different in single nucleotides. This is called, as I said already, single nucleotide polymorphism. And there is great hope that we are also differently radiosensitive because of our SNP map. So now with modern high throughput molecular biology methods, you can analyze SNPs in a GWAS approach quite easily. And there are several studies ongoing, large international studies, where a fingerprint of radiosensitivity or a biomarker of radiosensitivity at the level of SNPs is analyzed. Some of these studies were already published and they are promising. Some are ongoing, so we have to wait to see what comes out. But generally speaking, I think there is hope, and the results that we have are quite promising. Yes, it may be possible to find a fingerprint at the level of SNPs. It's going to be a different fingerprint for every side effect, for every cancer entity, and so on. But we will see. There is a lot of work going on, and that's very interesting, very promising. But there are major confounders that we have not overcome. And some of them are well known, and some of them, I think, are less well known. So one confounder that I usually don't find that people take account of is the difference in decontouring or contouring or delineation of organs and risks, or generally of the target that is supposed to be irradiated. Now, there are quite a number of publications where people analyzed. You see, what you do is you take treatment plans from different centers, or even from different radiation oncologists from the same center, and you put them against each other, and then you check how did the individual radiation oncologists delineate the organs at risk or delineate the target volume. And this is from one publication where you see that there are quite large differences. So there are different delineation procedures. And obviously, how can you, when you look at the side effects of patients that were treated differently, they were really treated differently for the same disease. How do you know that the outcome, or if some of them develop side effects, this is because they are radiosensitive? Isn't this because they were maybe planned in a different way? So this is this is a major problem that I think is often overlooked. Well, another problem is the dosimetry. We are now looking at side effects in patients that have been treated maybe 10, maybe 20 years ago, where the treatment planning systems were not as good as they are now. So you don't have precise dosimetric information. You don't have dose volume histograms, for example. So it's difficult really to assess. And then you would have to go look into the journal of every individual patient to really check what was the dose that he received to this particular site where he developed side effects. And then we also know that treatment planning systems are not very precise when it comes to doses which are distal to the target. So different treatment planning systems, make mistakes. And this also has to be, this is a confounder. This has to be taken account for. And then there are large differences in how different oncologists and how different centers evaluate side effects or adverse effects. However, here there are attempts to standardize things, and Catherine West, who maybe is in the audience, I don't know, but she had a lecture just in the morning in a different session. She is running a large project called REQUITE, and they are trying to standardize side effects just to get a grip on this, so that that you really are able to compare patients, which currently is difficult. It's not it's not so straightforward. You read a publication and you think, yeah, it's easy. You identify patients that develop side effects, and you identify patients that don't develop side effects. But what is a side effect? I mean, the patient that I showed you, this was extreme. This was really extreme. You don't have many patients like this. Usually you will have inconsistency at different grades, and how do you grade this? Okay, so that was for radiation sensitivity. Let's go and have a look at radiosusceptibility. Again, we assume that your genetic background is I think most of us assume this, is a strong factor that influences your radiosusceptibility. So, if that is the case, again, I can take blood from you and I can irradiate this blood under in vitro conditions, and I can rank it with respect to the level of radiation-induced chromosomal aberrations that I see. And then I will have some of you who have high levels of chromosomal aberrations. Lymphocytes were all exposed to the same dose. And then I will say, okay, you are at a high risk of developing cancer. I mean, if there is a correlation between in vitro sensitivity of your lymphocytes and your cancer susceptibility, then it should work. And it should work provided that this radiosusceptibility is genetically determined. However, if we look at publications which attempt, or studies which attempt to find out what is the component, the genetic component to your risk of developing cancer, spontaneous cancer, then the genetic part, this is from a study published in 2002 in Sweden. This was and the title of the study is Environmental and Heritable Causes of Cancer Among Nearly 10 million individuals in the Swedish family cancer databases. So you do family linkage studies to find out how many cancers are heritable or have a heritable trait, and then you can assume they are genetically determined. And there is a large variability with respect to different cancer types. So thyroid cancer, according to this study, is thought to be 50% of cancer cases is due to your genetic background. Endocrine glands, about 30%, testes, breast, 25%, and then we go down. Then you have some cancers where there is nearly no genetic component, or genetic component is very low. And recently there were two studies from Fogelstein and Tomasetti, or Tomasetti and Fogelstein, where they correlated, or they tried to find out what is the correlation between replication errors or number of stem cell divisions, assuming that, and we know actually that every time a cell divides, there is an error associated with DNA replication. So the more often a cell divides, the higher the risk that the cell will carry some mutations. And what Tomasetti did is he correlated the rate of cell divisions in stem cells with the risk of cancer. And he came to the conclusion that most cancers, approximately 70% of cancers, are due to mutations which are caused by replication errors. So there is no really a genetic component there. So the larger you are, the higher the risk of cancer, because the more, simply speaking, and in fact there is some data indicating, yes, the larger you are, the higher the risk of cancer, and vice versa. Okay, so this will just, I'm mentioning this to point out that the genetic component in cancer predisposition may not be very large. But now, there was a study that really kicked off many other studies in the 1980s, 1985, from the lab of Catherine Sanford, who was a very famous cell biologist, where they took fibroblasts from different cohorts and looked at the aberration frequency, radiation-induced aberration frequency in these fibroblasts. And they thought, well, maybe there would be a correlation between the radiosensitivity of the fibroblasts and the cancer phenotype if you irradiate cells in the G2 phase. I won't go into details why they did it, but there were some, of course there was a reason for this. And what they found was, what you have here is the number of aberrations per 100 cells, here on the y-axis, and then you have normal donors, you have donors with genetic disorders, which are cancer-prone, cancer patients, and then you have tumor cells. And you see that you have more individuals with a higher aberration score among these, in this cohort as compared to normal donors, and in this cohort as compared to normal donors, and of course in cancer cells. Cancer cells are genetically unstable, so this is what you would expect. The problem with this study is that the majority, or the highest effect, is seen at the level of these gray bars, and these are gaps. Now, I don't know how many cytogeneticists are in the room. I am a cytogeneticist, and I think most of us cytogeneticists agree that gaps, we generally don't score gaps because they are not so easily identifiable. So if you have any result at the level of gaps, you would say, maybe. We rather look for breaks, we look for exchanges. Gaps, maybe. So the problem here is you do see an effect at the level of breaks as well, but the major effect is at the level of gaps. So maybe. But then there is another set of studies that was carried out by Stefano Bonassi, who is working in Italy, and what he became interested in, and this is really a cool approach. These are really nice studies. So he thought, well, if imagine you find cancer patients, and imagine these cancer patients would have had their chromosomal aberration frequency studied 20 years ago. Then we could correlate the aberration frequency with the risk of developing cancer. So he set out in the world and he was looking for databases, and there are a number of databases where, like, people starting a job somewhere, they are asked to give their blood, and then their baseline level. We are talking about the spontaneous frequency of chromosomal aberrations, where the baseline level of chromosomal aberrations is studied. So he managed to get hold of a number of such databases, and then he went into cancer registries. He looked into cancer registries, and he identified then patients who developed cancer, and then since he had the database for the chromosomal aberration frequency, he could then correlate the level of chromosomal aberration frequency with the risk of developing cancer. And he published a number of studies. This is taken from a publication in 2008, and he plotted the results as, this is a Kaplan-Meier plot, for cancer-free probability as a function of time since the chromosomal aberration test. And he divided you see, the frequency of spontaneous chromosomal aberrations is rather low. You often have 1 or 2 aberrations per 100 cells or so. So it's not, you don't have high numbers. The statistics is tough. So what he did was he stratified the cohort into 3 groups: low aberration score, median aberration score, and high aberration score. And you see that there is a trend. Like these guys with a low aberration score, they tend to live longer, cancer-free, than these people here who had a high aberration score. And the median one, the medium group is in the middle somehow. So it fits really nicely. When then he looked at, well, it's the interesting question, of course, which cancer are we talking about? And then he found, well, the strongest association between a high aberration score and the cancer risk is for stomach cancer. And that may be linked to the metabolism of agents involved in stomach carcinogenesis, like high folic acid and vitamin B12. But it may also be linked to the fact that many people who have a Helicobacter pylori infection develop stomach cancer. And we know that people who have a bacterial infection, bacteria tend to excrete restriction enzymes, and they cause chromosomal aberrations in lymphocytes. So this outcome suggests, well, what are we looking at? Are we really looking at the genetic predisposition to cancer, or are we looking at the effect of maybe an infection which then led to cancer? I don't know. You see, this is, again, one of these problems where we say, okay, I mean, if you pronounce quickly, it sounds very nice, it looks very nice. Yeah, this is a straightforward result. The more aberrations you have, the higher risk of cancer, but maybe not. Okay. Lymphocytes of breast cancer patients were analyzed by David Scott. So I actually was at a meeting in 1989 where David Scott heard the results presented by Catherine Sanford. And he said, no way, this can't be true. David Scott was a cytogeneticist. And then he went to his lab and he started to do these studies. And he came up with results which actually quite confirmed the results of Catherine Sanford. He took lymphocytes for breast cancer patients. He irradiated them in vitro, and he used the so-called G2 assay, and he looked at the aberration score. Now, so what you have here is a frequency distribution of patients, or normal people, so healthy people, with respect to their aberration score induced by radiation. So this is radiation-induced aberrations. And you always have a distribution. You have some patients that have a high frequency of aberration. So he said, okay, these we make a cutoff and we treat these 10% as radiosensitive. And then you look at the cutoff, this is like 100 aberrations per 100 cells. And then we apply the same cutoff to the cancer patients. And he found that about 40% of cancer patients were more radiosensitive or showed a higher in vitro radiosensitivity as compared to 10% of normal healthy patients. So this would suggest, yeah, a high, I mean, it goes in the same direction as Kathleen Sanford's results, that high patients that have cancer have a higher risk or have a higher level of chromosomal aberrations or in vitro radiosensitivity. And then he looked at the question, is this heritable? So he identified the patients, so he took some patients from this group, and then he looked at their children. And 63% of the children had the same phenotype of high radiosensitivity. So it seems, yeah, there is some heritability here. And there are a number of studies where people looked at different cancer sites, and not all cancer sites show such high cutoff. I mean, for gynecological cancer, 16% sensitive, but not so many patients. For prostate cancer, 50%. For head and neck cancer, about 40%. But there is something there. But the strange thing is that we see many more. I mean, you would expect, and yeah, this in vitro radiosensitivity, this is a genetic trait. But this is the percentage of people that show this high radiosensitivity is higher than the genetic component that you expect to contribute to cancer. And in fact, there is a paper by Wu who has been analyzing sensitivity to bleomycin as a marker of high sensitivity or susceptibility to different cancerogens. And he says, well, about 60%, the genetic contribution to this high in vitro radiosensitivity component is about 60% genetically determined. So that's much higher than what we know from genetic linkage studies. Why? How does this fit together? And then there are studies where, like this study from Hungary, where they looked at high radiosensitivity of lymphocytes from cancer patients and healthy people, and then healthy alcoholics, who obviously are quite resistant, because if you drink a lot, if you smoke a lot, and you don't develop cancer, then you are quite radiosensitive. And he didn't find any correlation. Well, he found that the alcoholics had the most aberration. So, okay, my time is running out. Then you have studies like this one, where people find that women that are left-handed have a higher aberration score in their lymphocytes. They're more radiosensitive. What do you do with an outcome like this? Would you say, okay, oh, sorry, lady, you're left-handed, I am not allowed to employ you in the radiation protection area because you obviously have a higher risk of developing cancer? Would you do this? I doubt it. Okay, I will skip this. There is a lot of interest in second malignant neoplasms, because we know that people that have been, especially children that were treated for cancer in childhood, they have a high risk of developing cancer. And then maybe they are particularly radiosusceptible. The problem is, though, that we don't really know what is the dose-response relationship for cancer induction. Here we disagree a little bit within our working group, but I'm showing you a paper that was published in 2009, where they try to localize, they try to map the dose to the tissue where you had second malignant neoplasms. And yeah, it's kind of difficult to say, yes, definitely you cannot say there is a dose response. If there is anything, then there is a reverse dose response. The lower the dose, this is the dose to the site. We don't know. And then there are problems also with dosimetry that I mentioned already. This is from a study where they looked at the dose to the stomach following radiotherapy for Hodgkin disease. And depending on the type of treatment, you have different doses to different parts of the stomach. How do you report the dose? If you take the average dose, it's meaningless. So again, dosimetry is difficult here. Now, again, I said that genetics, we all assume that it's genetics. It's our genetic susceptibility to cancer. But Actually, Professor Niwa, who is here in the audience, at the last day of the ICRR meeting in Kyoto, he gave a lecture where he pointed out that we can influence our radio susceptibility. And I think this is extremely important to mention. I mean, all risks are conditional. So you can actually, by changing your lifestyle, you can you can modulate your risk of cancer even after you're exposed to radiation. And this is extremely important. I think this is a message, well, this is something we want to look into, but we think this is important because then we know what has happened. We know of the psychological effects after the Fukushima accident and the Chernobyl accident, of course. And if you convey this message to the people that they can actually control their risk, this is going to have an enormous implication for their well-being, because suddenly they are in control of the risk. I think this seems to be very important. So they can modify after radiation exposure the radio susceptibility. And there are, of course, there are many studies that show that your lifestyle has an influence on your radio susceptibility. So the conclusions, I will make it. I have 30 more seconds. Functionality assays to detect individual radio sensitivity are not really, they don't give good results. So they got conflicting results, and yeah, their value is doubtful. Radio sensitivity is a complex trait, so SNP analysis by GWAS is very promising. We should wait for the outcome of these large studies that are ongoing. There are a number of confounders when looking at radio sensitivity, like variability in contouring, organs and risk, and defining adverse effects, and these need to be overcome. The value of testing for radio susceptibility in the context of radiological protection of low dose among occupationally exposed individuals is doubtful because of the low contribution of genetic background. And the effect of lifestyle and other factors on risk following radiation exposure, so-called effect modifiers, should, we think, be looked into better, should be understood better, so that you can then take control of your risk after you have been exposed to radiation. Thank you very much.